So in this tutorial, we're going to go over how to use the PantheonOS platform for single-cell genomic analysis. So before I begin, the first thing I want to mention is, so first I'm on the online platform. If you're using the desktop app, it's the same exact steps, so it's very translatable. And before I do anything, one thing I want to specify is this first page here, where I can start a new conversation with the platform. Because I'm doing single cell analysis specifically, I can click the setup tab in the bottom of the chat box, and then I can select a team of agents that work very specifically for single cell analysis. So I can select the single cell analysis team. If you want a team that's very specialized towards single cell genomic analysis, spatial transcriptomics, this is a great option. For the purposes of this tutorial, I'm just going to stick with the general team by just giving a heads up that option. It's a great option if you're focusing specifically on single cell analysis. So obviously the first thing that I want to do is I want to attach a data set. So I'm going to click on the paperclip in the bottom half of this chat box right next to setup, and then I'm going to pass on a file. And obviously it's a very large file, so I'm going to wait a couple minutes for it to load. So as you can see, after a couple minutes, the file is finally attached. And now I am going to give the platform a query. So I'm going to type in the chat box. Before giving any prompt, it's always best to kind of just very briefly provide context for what the file you've attached is. And this is especially true if you're attaching multiple files, especially for something like spatial transcriptomics. So this is a genomic data set taken from a healthy Okay, so this is a relatively simple prompt. You can make your prompts as general as you want. I mean, this is pretty general. You can make it as specific as you want. You can also make the prompts a lot more complex. You can specify that you want a complete workup on a data set. So for example, I could specify that I want quality control, normalization, dimensionality reduction, and then differential gene expression analysis, especially if I had, say, another data set, like for example, a disease data set that I want to compare with. You can really do anything with these prompts. And you can also specify. So for example, I could specify that I want this done in a Seurat pipeline, or I could specify the specific thresholds that I want for quality control. You can specify exactly what kind of steps you want the platform to take, exactly what kind of result that you want, how comprehensive you want the workflow. It really is open-ended. I am just kind of providing a more general, simple prompt for the sake of this tutorial. So I'm going to enter this, hit enter. And then as you can see, I'm getting a notification that the agent is going to start to respond. So what I want to point to immediately is, so when I go to the top right corner, I click the side panel icon, and then it pulls up a side panel. So in this side panel, as you can see, we're already getting notifications of how this platform is thinking, if you will. So this timeline tab shows you all the steps that the platform is taking. And as you can see, it's going through them right now. And then it'll provide kind of a summary of those steps in this main panel here above the chat box. The platform will also create a plan of all the steps that it wants to take, and that can be viewed in the Plan and To Do tab. So as you can see, the plan is right here. And then any files that are outputted at any time will pop up on the Output tab, which I can view here. So after a couple minutes, you can see that the analysis is complete. So you get the notification in the main panel, and then you also get the notification in kind of the side panel. If you go to the Plan and To Do tab on the side panel, you can see all the steps are checked off. If you go to the Output tab on the side panel, you can kind of see the outputted files, and then you can click through them. So you can click on the figure of the most variable genes. There's both a volcano plot and a bar graph. And then I can go back. Once I've clicked a file, I can go back by clicking the purple arrow next to the name of the file. There's a CSV file with the top 100 most variably expressed genes. And at any point for any of these files, I can also click download, this icon over here. I can expand the preview with this icon, and then I can also edit the file if I want to with edit mode. But the other thing I want to show is the analysis notebook that's outputted. So this is basically all the code. This is the algorithm itself that is used to do all the data analysis and output the results. This notebook is interactive, so you can use it as you would in Jupyter Notebook. You can add cells, you can edit the code, you can run cells however you choose, and you can essentially manually alter the analysis that is carried out by the platform. So this is very interactive. The other thing that I want to kind of show is, so if I close the side panel, if I click the 3 lines below the Pantheon icon here on the left-hand side, and then if I click the folder icon, you can see how the project is grouped into a folder system as well. So the .uploaded files, it includes the file that you uploaded, and the output for this project is registered in the Fetal Heart HVG folder. It's just the name the platform automatically gave the folder. And then you can see the one figure. Any figures that are outputted are going to be grouped in the figures folder. And I can click on the figure and it opens the side panel to show the figure. Then there's also the analysis notebook, and then the outputted CSV file, which is also important. So yeah, I just wanted to show that file system. And obviously, files that are kind of to the platform itself are going to be in the .pantheon folder. Those files aren't really, this folder is not something you should really worry about. Yeah, this is just a brief overview of how to kind of genomic analysis with the PantheonOS platform. Obviously, you can expand this to spatial transcriptomics by multiple files. So not only a genomic data set, but Visium mapping, then the images themselves, and any imaging mapping files. Don't specify what each file does. That's okay. The platform will look at the files individually and kind of what file does what. But it's always easier to kind of in the prompt itself. And then you want spatial transcriptomic analysis and any specifications of what exactly you want, specific workflows that you want. Yeah, very open-ended, and it will give you rigorous results. Yeah, it's just a brief overview on how to do genomic analysis with the PantheonOS platform, and we'll see you in the next one. Thank you.